Hello everyone. Today I am going to present my PhD thesis work, which is just for the recording purpose. And this thesis work mainly focuses on signal transduction, pathways and the networks, mathematical modeling, simulation, and implementation of the algorithm for the simulation of signaling pathways and the networks. So for basic introduction, I have selected the TCR mediated MAP kinase signaling because my model is based on MAP kinase signaling pathway. So since I'm using mathematical modeling, so it is mostly related to mathematical models. So I wanted to introduce one biological pathway just to understand better those people who do not know much about system biology and mathematical modeling. For them, it will be easier to understand. So in general, when we are talking about a signaling pathway, it starts from receptor, which receives the input signal to trigger the internal signaling process. That means the cell signaling. And then finally, it reaches the information or signal to the nucleus. Similarly, here, TCR is one of the well-known receptor. And what we are showing here that TCR is activated once it receives the input signal. And then LCKs, F70s are involved, which basically also involves grab2, SOS, LET, and PLC gamma. Then it goes to the RAS GDP, which is like inactive form of RAS, which is converted to RAS GTP, and then which is like the RAS GTP is the active form of RAS. Then RAS GTP activates RAF, which is activated after phosphorylation. Once RAF is activated, then it is further activating MEC by phosphorylating in single place or on double place. That means single phosphorylation or double phosphorylation. Then active MEC, that means single phospho form of MEC or double phospho form of MEC, could activate further ERK to single phospho form or double phospho form. RAF could directly also activate ERK in the similar way as MECP and MECPP is doing. In general, single phosphorylated MEC is also called as partially phosphorylated or partially active. Double phospho form is called as fully active. Now, once this is activated, so basically we consider on the final molecule which is located in the downstream of this pathway, that is ERK. So how these fully activated ERK behaves with respect to time. That means what is the response, how the temporal response changes. So that is what in general is considered as one kind of indication for the cells undergoing some kind of changes like proliferation, apoptosis, cell growth, differentiation. That is also, we call it as cellular phenotype.